Okay, Carl, if you want to go ahead with the intro, we can start.
Okay, sure. So, hello everyone. My name's Carl Wegman. I'm a fellow here in the Center for Geospatial Analytics, and it's my pleasure to introduce to the geospatial community my colleague and friend, Dr. Chris Osburn. Chris is an associate professor in marine biogeochemistry in the Department of Marine Earth and Atmospheric Sciences. He's a fellow in the Center for Geospatial Analytics and recently was named a university scholar here at NC State University. Chris's academic career began at Indiana University, where he earned a bachelor's degree first in public affairs, followed a couple years later by a second degree in geological sciences. In 2000, Chris earned his PhD in earth and environmental sciences at Lehigh University, an awesome institution I know something about. Before joining the faculty at NC State in 2008, Chris pursued estuarine and marine-based environmental science efforts as a research chemist with the US Naval Research Laboratory in Washington, D.C. Chris's research interests and the successes that his group has had focused primarily upon the biogeochemistry of organic matter transferred from the land to the ocean, emphasizing how extreme weather events like hurricanes, which I think we'll hear about today, and how human activities like the Deepwater Horizon oil spill impact coastal water quality and carbon cycling. His research efforts are funded by NSF, NASA, the US Environmental Protection Agency, the Office of Naval Research, and the North Carolina Sea Grant and Water Resource Research Institute of North Carolina, among others. He's widely published and cited, including in the journals of Environmental Science and Technology, Biogeochemistry, Marine Chemistry, PLOS One, and one of my favorite go-to journals, Journal of Foraminiferal Research. Chris is at the forefront of chemical and optical analytical approaches to studying the flux and fate, that's better, of dissolved and particular organic matter, microbial organic matter interactions, and the implications of remote sensing of surface waters that include terrestrial, estuarine, and open ocean settings, and span the hemisphere from the tropics to polar latitudes. His research includes studies in disparate biogeochemical settings, including lakes that are formed as a result of glacial retreat in southwestern Greenland, the Costa Rican rainforests, streams that derive portions of their chemical composition from the degassing of magma beneath active volcanoes, the Hawaiian estuaries impacted by the legacy of US naval operations. He has tracked the fate of organic carbon from the Florida Keys and the Gulf of Mexico, stretching from Corpus Christi Bay in Texas eastward to the Apalachicola Bay in the Panhandle of Florida. Moreover, of course, in North Carolina, Chris has active research spanning spatial scales from small estuary creeks near Bald Head Island to the much larger Neuse River system, its estuary in Pamlico Sound. Please help me in welcoming Chris Osburn today as he presents a seminar titled Geospatial Analysis, Coastal Water Quality, and Carbon Cycling, a Biogeochemist's Perspective.
Well, Carl, thank you very much.
You're welcome.
Can I have a copy of that? Because
sure, I'll email it to you.
Thank you. No, it's definitely an honor to be here. I'm happy to be a fellow within the center and to finally get to present some of my research and my perspective really on how a biogeochemist like myself can try to interact with other folks within the center and other scientists and integrate geospatial analysis in our more focused aspects and studies we do on coastal water quality and carbon cycling. So this is a perspective talk. And I want to acknowledge, Carl's mentioned some of these already, but I do want to acknowledge the specific things that we'll get into today and some of the funding sources for those. And especially NSF chemical oceanography, and then local support from NC Diener and some others here. And also acknowledge scholarly reassignment. We're at some time to kind of, in Denmark actually, sit down and think a lot about some of these topics and try to pull some of these ideas together. And of course, I also have to acknowledge my list of colleagues and collaborators here who have really provided a lot of good counsel and interactions in thinking about how we can merge these topics together and provide some perspective for all of that. And another thing I'd like to point out is that what I'm going to try to do today is to highlight aspects of my research group, and specifically projects that present and former students have been working on, and postdocs. And so we won't touch on everything, but we'll get to a lot of I think really exciting types of things that we have done and we are doing and we hope to do in the future. And so this overview here gives us an idea of where we're going. And so what I'm hoping to do today is to talk about how coastal interfaces are these dynamic environments, and they're places where there's active carbon and nitrogen cycling. It makes it challenging for observationalists, but also makes it challenging for modelers to try to understand how to represent these interfaces in things like Earth system models. And so I'm going to also then kind of make this point that the more classic ways that we've looked at how these interfaces behave and the ways we've been able to represent them will benefit and will make more progress from taking this kind of wider geospatial context. And so I'm going to pull together a couple of things that we've done and give us a sense for how we can use this approach to understand these systems, these interfaces, but then also what some of the challenges have been. And so from that, I'm going to pull on about 10 years of research I've done here in North Carolina, with going elsewhere a little bit with some other associated and future studies. So how do you tackle this type of activity? Well, one way is to sort of focus in on one aspect of carbon and nitrogen cycling, and that's organic matter. And so we're going to go over a little bit of background on why dissolved organics matter, what their role is in the coastal carbon cycle, what these interfaces are, how do we define them, and how are they represented? And then as to build in some of the things that we've done in looking at how presses such as land use and pulses such as extreme weather, hurricanes, tropical cyclones, and all that, how do they alter the carbon cycle, and how do they, or the coastal carbon cycle, and how do they actually make this more challenging, and again, benefit from taking this more of a geospatial context for understanding these roles. So let's just take a quick trip to see why dissolved organics matter. So there's a lot of vernacular that kind of means the same thing, but they're used in different ways. And so when we talk about dissolved organic matter, we quantify it as dissolved organic carbon most of the time. And this material is consumed by heterotrophs. And you can see here dissolved organic carbon going up through the planktonic food web into the higher trophic levels. And it's a basal resource. So one of the important things about DOM is that organisms eat it. And so it forms this kind of basal supply of carbon and energy into food webs. Another aspect of it is that it directly influences the chemistry. So some of these molecules contribute to acidity, other molecules contribute to alkalinity. And it serves this purpose, some components of DOM serve this purpose to act as complexing agents. And so they can bind up metals and phosphorus and hydrophobic molecules. So the organic matter itself can be important vectors for transport. Just a quick example of that, this graph here on the right showing the absorption of the contaminant phenanthrene onto humic acid, which is a type of dissolved organic matter, in the presence of ions and minerals. And then finally, near and dear to my heart, and also important for our geospatial context, this stuff absorbs sunlight. Not all of it, but a large fraction of it does. And so we call this CDOM for dissolved organic matter. And you can see kind of the dynamics here in this little cartoon. And we'll talk a bit about CDOM. And so to give you a sense for the kind of the variability in what CDOM is, a lot of people don't know that term. I have this stock image that I've used for years and years now that was taken from an overflight of the Charlotte Harbor in the Gulf Coast of Florida. And what you can see here is this nice brown plume flowing out of the inland back bay here onto the shelf. So out of the outflow from Charlotte Harbor onto the West Florida shelf. And this brown material is the CDOM. It's largely coming from land, and you can see here it's bifurcating or flowing through this nice little green CDOM. That is organic matter, color dissolved organic matter from algae. And so we see that the optical properties here are important and can be used as discriminants of material coming from land and material formed in coastal waters. And so because of that, the CDOM ends up being an important tracer, and it's one of the things that my lab's been working on for years and years now, and something that we'll get into in today's discussion. So the characteristics of this material, and trying to put it in context of carbon and nitrogen cycling, the dissolved means that it's less than about a micron in size, and so we can just use simple filtration to actually collect that from a water sample. You can see here in this plot, this little cartoon, that DOC makes up in the ocean about 700 gigatons of carbon, so it's a very large reservoir. There are large sources of it on land here, and both in some of the more geochemical reservoirs and biochemical reservoirs. And it acts as this major conduit from land to ocean because it's soluble and it's easily transported in streams and rivers. Might be stored in lakes or in wetlands for a period of time, but then exported onto the coastal or into coastal environments. And you can see that DOC specifically constitutes a very large flux, or very large yield in this case, in terms of the amount that's delivered from rivers to estuaries and ocean margins annually. So we've got this material, and you can see that it's a large magnitude, but another thing that I'd like to point out is that it also has a variety of sources. And so when you get in the DOC game and you start looking at this, we have to think about not just the type of material the concentration that's there, but also the type of material. And so its quality also matters. So just as an example of that, we're beginning to understand a little bit more about how these different components of the surface system can contribute varying amounts of this DOC to streams and then rivers and then ultimately into coastal ocean. And so I've got a couple of examples of this with respect to wetlands, and I'm throwing wetlands out here now because this is going to be an undercurrent that we're going to come back to and think about as far as how the spatial context can help us understand more about how these coastal interfaces work. So to kind of build up that idea, let's first look at this plot here, which shows how DOC concentrations increase as a function of wetland, percent wetland area in a catchment. And I'm showing this study from Gorham and others in 1988. These are just small streams in Nova Scotia. They noticed a lot of seasonal variability, but they also noticed that the color to DOC ratio increased with this increasing wetland influence. And so the wetlands themselves are sources of the CDOM as well as this DOC. So the quality matters, the quality is changing. What also turns out in larger systems that wetlands contribute quite a bit of DOC to larger coastal rivers, such as the Neuse River in North Carolina, Cape Fear River, and one large system such as the Mississippi or some of these others. So that's just shown here too. Again, looking at this CDOM yield as a function of the percent wetland area. And so the idea here is that we can have this sort of variability that we'll begin to see in that freshwater end member coming in across that interface. And that's something that we're going to try to work on and understand a little bit more in detail. Okay, so that gives us some background as to what DOC is and to some of the considerations that we can think about in a spatial sense. How do we study it? Well, we can go out, as you can see here in these pictures, and we can collect it in a number of different ways. We can collect it from boats. We do a lot of collecting from bridge overpasses here. And as I mentioned before, the distinctions based on size, so we can filter it, take it back to the lab, and then do our analyses. So what we typically do in our group is we measure light absorption. You can see that represented here on an instrument called a spectrophotometer. We can measure the DOC concentration. You've seen a couple of examples of that. Then we can also measure the stable isotope value. So what the isotope values do is they give us another dimension on the molecular level that we can compare to some of these other measures. And so I mentioned that not just measuring the concentration is important, but also looking at the quality. And so in combination, we can derive a lot of different metrics off of these measured properties in order to look at that aspect of quality. And so one that we'll talk about today is the SUVA value, which is specific UV absorbance at 254 nanometers. What does that mean? You don't really have to worry about what that means other than it is a amount of light that's absorbed divided by the DOC concentration. And most importantly is that it tends to increase in value with the aromaticity of the organic matter. Now, aromaticity means aromatic rings, and those are specific carbon molecules that tell us that it's from a source enriched in humic substances. And would be prevalent and prominent in environments such as wetlands. So we've got a tracer that we can use called SUVA here. Another tracer is the SR, which is a ratio of how the light changes over wavelength. And these are just slope values. So we calculate, log transform, calculate these little slopes. The nice thing about doing that is that there's a relationship between the SR value and the molecular weight of the material. And so like having a lot of aromaticity, higher molecular weight material with specific SR values we can use to distinguish are consistent in different environments such as wetlands versus main river channels versus these coastal areas. And then finally with the DOC stable isotopes, you can see here we've got different ranges of these values that we can use. Now what the stable isotopes do is they look at how the 13, that slightly heavier isotope, partitions into different materials based on biochemical, geochemical processes. So what this means is we've got a number of different tracers that we can use to track the movement and sources of this material through the system and begin to unravel how those sources play out. So again, it's not just the concentration, it's also looking at the quality of this material. So that gives us a perspective on how to analyze the carbon part of the story here. How do we put this together with interfaces, and what exactly is an interface? So I've got a few ideas or a few things pulled together here to kind of set the stage for that. And one really comes from this cartoon here on the left, which shows how these interfaces are sort of represented in Earth system models and how they're actually represented in a very straightforward and poorly, in a way that has poor resolution to it. And so what I mean by that is that the Earth system models have a connection between the land and the river, but the river is basically seen as a pipe that just takes and delivers material into the ocean. Well, we know that it's much more complex than that. We have a lot of features to the landscape that are not very well represented in these models. And so the interface can really exist across the entirety of the transition from land into the ocean. So we can think in a matter of scale, from very small scales, say from groundwater to surface water, or even pore waters within the subsurface exchanging with overlying waters, as much as we can talk about these interfaces being very large transitions of rivers into the ocean through estuaries and so on and so forth. So we have a number of different ways we can define these interfaces, and what I'm going to do is sort of narrow that down to the estuarine perspective for this talk. I've lost control of my, it's still remaining. Okay, so DOC in the coastal carbon cycle, then we've got this, across this interface, we have a lot of different types of processing that can go on. And so one of the important things to point out here is this material tends to move through the system. A great deal of it can be reacted and eventually create CO2. So again, when we look at this interface, we have to think not about just the transfer of sources, but also their reactivity and how they move around. And so a geospatial perspective on the carbon cycle could be represented in a couple of different ways. And one is with this sort of conceptual model, this cartoon here that you can see, where we look at the changes over a change in landscape. But then we could also be quite a bit more sophisticated with this. And this is from a study by Yang and others, where what they did is they integrated DOC concentration data from monitoring networks across the US, and they're able to then regionalize concentrations based on breaking it up into different watershed units. And so this is from the kind of the delivery system end, sort of the where a lot of limnologists and river scientists are in terms of integrating these land use and these regional properties, the spatial properties, with trying to then begin to understand how that influences the quality of material that's delivered to the system. And so a biogeochemist perspective on this might look at it, but flip-flop it kind of the opposite way. So we're not necessarily taking the perspective of from the spatial end and then looking at the output. We're trying to understand how what we see in the output in the laboratory reflects back to the starting material, or sorry, the starting point of the system rather. And so, this little graphic here describes how we might be able to think as transitions in, say, a watershed, the land use watershed from urban forested into agriculture and into wetland, how that influences these properties we can measure, such as the molecular weight of the DOM, and then because of the metrics we have, we can hypothesize about how those should play out and look at the response functions in that sense. Another way of looking at this is to try to view this in terms of changes in different sources as we move throughout a coastal system here, as you can see represented on the right. So I'll get into a little bit more detail about what these figures correspond to here. But just to say that, just to kind of show you, I think, two perspectives on how we can look at the same thing. So we have the spatial context, and we have the chemical means of trying to pull apart all of these different signals. How do these presses and pulses end up altering the coastal carbon cycle? And so first thing to think is, what do I mean by presses and pulses? These are ways of talking about disturbances. And so it goes like this. So a pulse is something like an episodic or acute event. And so extreme events like tropical storms or hurricanes, these are episodic and acute pulses that are going to disturb our interface, disturb our system. Contrast with that are gradual and chronic presses. And so these are changes that might be occurring at a much slower rate and perhaps even imperceptibly, but they're occurring and they're sort of pressing on that interface over time. So that interface might be moving and the dynamics of it might be changing over a sort of a shallow or slow gradual perspective, but also this kind of intense perspective from these events. We care about describing these because what they do is they integrate environmental and social domains. And so the events tend to disturb or disrupt these systems. And along the way of how they're being disrupted or disturbed, they're being disturbed within these kind of long-term changes that we see. And I can point out too that integrating these domains together with both the environmental, the biogeochemical, the water quality, and the social domains, what this ends up doing is that this could actually have implications for what's termed the blue economy. And so the economy related to the activity of the coastal activities within the coastal environment. And so I bring this up to say that there are some larger perspectives and connections that we can make at understanding how carbon and nitrogen cycles, the biogeochemistry, manifests in water quality and makes connections to much larger things that we might be interested in. And then just as some quick examples of this, these event pulses can initiate feedbacks that alter coastal communities. So strengthen, again, that connection between how the environment works and then some of the results in terms of flooding, 2 examples of flooding that you can see here, and then in terms of water quality. Now, again, with that, the other thing to bear in mind is that these pulses can occur under presses of land use change that are changing the source inputs of organic matter. So I'm going to loop back to this figure that I showed earlier. This is a paper we did a couple of years ago on this, where we actually used a technique related for measuring CDOM called fluorescence. And we were able to show that the quality of organic matter in the more urbanized upper part of the Neuse River Basin here is very different than in the agriculturalized and wetland-rich lower basin. And so we'll kind of visit this in a little bit more detail in just a second. Now, with these presses of land use change and these pulses of extreme weather that we're seeing, these events, this creates, again, this sort of conundrum here, because we now have a much more dynamic system, one that's harder to predict. And so how that coastal interface is responding to increased storm events and overall increased precipitation and flooding, these are all things that are requiring us to really go outside and integrate in a much broader spatial context for understanding how the biogeochemistry is operating. Now, of course, it turns out that North Carolina is what we call it in the biogeochemical realm, a hot spot experiencing a hot moment. And so for better or for worse, it happens to be a great place to study these phenomena, because it tends to be getting wetter here, as well as we're experiencing more of these more frequent extreme or severe weather events and storms. And so what we've begun to understand from these is that these events, these pulses, dramatically change how we view the interface and how we can actually model the interface. And one of the big things that we've learned over the last few years is that a majority of the carbon and nutrients and other materials can be exported across the interface in just a few of these events. And that's what we're showing here in this figure. And so you can see that as far as the storm total as a fraction of the annual total, some of these years in which when we had these major events, the majority of nitrogen, the majority of dissolved organic carbon, and even inorganic carbon, large amounts of that's coming out in just a few of these events. And this ends up having a problem, or not so much a problem, but it can alter the ecosystem state of these receiving waters. And what I mean by that is that once you imbalance this interface, or you change this interface around, you change the biogeochemical cycling, what we've discovered is that this can shift the ecosystem state. Okay, so that creates a problem in the case where we want to be able to represent that system as perhaps a CO2 sink, but it is a CO2 source. And so this kind of leads us to this larger issue that I've been trying to think about, and how to integrate a geospatial perspective given the limitations of how these interfaces are often represented by coastal scientists. And one of the things that was done is, when you first start learning this, is you make plots of properties versus salinity. And so this is the way that we represent the transition from land into water. We have a fixed end member on the freshwater side, and we have a fixed end member on the marine side, or the seawater side. And if we assume that there's nothing but mixing going on between the freshwater and the seawater, then we can expect a straight line as we move, graphically here, move from onshore to offshore. And then deviations from that line, you see the dotted line above and the dotted line below, these would be considered as non-conservative. And so part of the problem with that is that it just presupposes that there are only two things mixing here. And pretty much we know that that's not the case, let alone we've got this additional reactivity that's occurring. And so one of the issues is that some of the patterns that have been reported just as a simple function of binary, two in-member mixing, that have been considered to be non-conservative might actually be conservative. And so what that means is that there's ways of constraining this end member, this freshwater end member, that aren't being taken into consideration with this more simplistic view. In addition to what is exactly non-conservative, and how do we make those interpretations? And so the simplistic view really narrows our focus. And I want to show a couple examples about how we can see the spatial variability due to these otherwise non-conservative, seemingly non-conservative processes. And so the first one here is looking at the effect of on nitrogen concentrations. And these data are from the ModMon program. And so this map here just shows the stations that are represented on the figure. So the same 11 stations are being sampled each time. And you'll see some seasonal variability between January and then April. But note here that in August, the yellow dots and then the white dots before and after Hurricane Irene, we see a massive drawdown of nitrate and nitrate, so inorganic nitrogen in this case. But the other thing to notice is that there's a shift in the distribution of these points. So that plot against salinity would not represent the interface in the same way, even though we've gone through and looked spatially at the same, we've revisited the same sites here. So you can see this variability in the spatial distribution that's in terms of where the interface is, and then the effects on, in this case, the nitrogen. Just another example here, kind of trying to finish this point of how the variability in the riverine end member is another source of this variability. And it means that just these simple plots against salinity aren't going to do what we need it to do here. And these are just three examples looking at CDOM concentration, DOC, and then lignin, which is a terrestrial biomarker, across a variety of estuarine gradients. So hopefully you get a sense of what our quandary is here, and then how we can better represent and connect together this idea of variability of what's coming into the system, and then some of the constraining some of the variability of the system itself. And so we're going to look at some examples here about how we try to tackle this over the really over a spatial gradient itself, as well as trying to develop some of these ideas. And so one of the first things I want to highlight here is looking at the changes in the source inputs. And I'm going to talk about a couple of studies that were done. First, the effects of land use. And this is a paper that Ruchi Bhattacharya did just recently, and she was a postdoc in the lab a couple of years ago. And so what Ruchi did is she started pulling together this spatial data for what the watershed looks like in the Neuse River Basin. You can see that here. The color coding are all the different observation points we had within the basin. And one of the things that she was interested in is trying to look at how some of these spatial variables influence the quality of the DOM. And in taking this into account, one of the major things that I want to draw your attention to is the transition of the urbanized and wetland percentages of the watershed as you move from the Raleigh area down into the lower coastal plain near where the river becomes its estuary. And so she did an RDA, a redundancy analysis on these data. And what she was able to determine is that, in fact, as you transition into the lower coastal plain, the wetland contribution was a major driver in terms of the quality, evidenced here by, here's our SUVA value, A254, DOC concentration, and even this value Hicks, which is another metric for humic substances. That this wetland feature was really driving that that change in the properties here. Whereas in the urbanized upper part of the watershed, it was mainly driven by slope and less of an influence of forested land use. And so this transition from upstream, more urbanized, to downstream, more agriculturalized is what we sort of expected, but we begin to see the wetlands pop out here as a major control on the organic matter, the DOC quality in the Neuse River basin. And so with that, you can also see how this is manifested in terms of both the carbon and the nitrogen as well. And the way that this plot's laid out here is that this is the RDA axis. And in these more negative values, where the wetlands were a larger contribution, we had significantly higher amounts of carbon relative to nitrogen. So changing that quality of land use ends up changing the quality of what's being delivered to the estuary. And so that had got us, doing that work had got us thinking about what actually goes on within the watershed might actually be another part of these major drivers. And so we got interested in looking specifically at altering the hydrology, and how does changing the hydrology change, say, the carbon cycle in these systems. An example of that is what you can see here in this plot, and this is a study that's being led by Stephen Richardson from our lab group. This is Wake County, and what we've got here, the blue are water bodies. You can see this is Falls Lake, Neuse River, and then some of these other impoundments that Stephen studied. We're going to focus on Sunset Lake as just a quick example. But consider that we're changing this landscape by storing water in all these various places, all these amenity ponds and other types of things. How does that change the carbon cycle? And so that's kind of what Stephen was looking at. So what he did was he did a transect around or down this Sunset Lake in Holly Springs, and it transitions from the water input here in Basal Creek. He's got points across and then to the dam spillway. So he looked at the optical properties and other properties of the organic matter, and the idea was to see if it's just consistent. Should it be the same? We might hypothesize that it's a pretty small water body. It's probably pretty well mixed. Does it all look the same? And what he found was there's actually a gradient that reflected this idea of how land use changes even over a very small system. So this is really very interesting stuff in the sense that you can see how dynamic these systems are. So in other words, what we see is that in the upper part, Basal Creek, that's more influenced by and expresses more of a wetland characteristic to it. You go back here and you look at the input here, you can imagine there's a lot of these sort of forested environments and wetlands here that's feeding into this lake around the stream that feeds into it. And then as you transition into towards that dam, towards the spillway, it actually shifts, the quality shifts into more of a planktonic system. And you can see that here represented in this picture that's showing this kind of algal bloom atop on the surface waters near the dam spillway. And so we've talked about reactivity, and I want to just bring up this point about reactivity again because it gets us into thinking about how what sort of the outcome could be for these systems. And so what Stephen did is he looked at the partial pressure of CO2, and the idea is if the PCO2 of the system is greater than that of the atmosphere, the pond's acting as a source, and if it's less than the atmosphere, it's acting as a sink. Again, thinking that, well, it's a pretty small system, maybe it's going to be very consistent. When he looked over time, what he saw is that there's a change that occurred that you could attribute to a rain event that basically we think mixed up the lake and kind of degassed it. And so you can see it's very well supersaturated with respect to the atmosphere. So the red lines representing the atmosphere here. But down where that algal bloom was occurring before the rain event, it actually did act as a sink. And so it's a dynamic system. And one of the interesting things about this project is I think that this is just got geospatial analysis written all over it. And in fact, what he's trying to do is to take some of these chemical measurements and then upscale them for the whole of Wake County using GIS and some other techniques there. So these perspectives on inland processes started to inform us really where of what could be happening with the spatial variability of these interfaces. So in terms of where the interface is, what's happening at the interface, and how these presses and pulses can influence carbon and nitrogen cycling. And so as we begin to truly move into the interface and think about what's happening there, I'm going to talk about two events that occurred in North Carolina, and I'm going to kind of shift gears entirely and talk a little bit about seagrass storage in Florida, because I think it also brings in another nice perspective on looking at geospatial analysis and thinking about using that to understand the interface. So we're going to continue on in the news here, and we're going to go back to 2011 to Hurricane Irene, because this is really the start of where I begin to think about how these storms can act as these major pulse events, but then also how limited we can be in trying to understand the nature of the quality of the, or nature and quality of the organic matter that's being transmitted from land to ocean through these events. So to kind of bring this together and to show you where our thinking went with this particular, the outcome of this particular study is that we did see, of course, a large increase in the amount of carbon that's being transmitted, but then these qualitative measurements, the C to N ratio and the SUVA value, all show that we had some pretty substantial changes to the quality that occurred because of the storm. So we're not just throwing in more DOC of all the same type, it's actually changing. And then we went and used another technique, this fluorescence technique, and we created with it these fingerprints that allowed us to look at differences in, say, C1, which is this component here for wetland sources, and compare that to kind of main river sources, C2, and then these more plankton sources here. And so what we're able to do is to kind of pull apart the organic matter in a little bit more detail and to look at the patterns of change. And so this is what the estuary looked like. So we're looking at the Neuse River Estuary here. And this is the upstream part, the river part, and then out towards the mouth where the river flows into Pamlico Sound. This is 3 days before Irene, so you can see salinity is increasing. And by and large, most of the estuary is sort of dominated from this upland river signal. After the storm, so after the landscape's been flooded, the effect of that is to move more material and have a very clear signal of more of a wetland signal that occurred. And this is 17 days after Irene. So we begin to use these chemical techniques to understand how the inputs are changing. And we're seeing that it has a strong effect of this wetland connectivity. And so what we learned from Irene is that these organic matter quality is shifting dramatically. But then we begin to think that it's likely coming from activating different parts of the watershed. So, Rucci's perspective gave us this idea that, okay, the watershed's changing and consequently that's, driving different properties. But now we're thinking that, it depends on where the rain's coming or where the water's coming from, where the rain's falling, that's driving that DOM into the coastal waters. But we do need a better geospatial way of looking at this. And so I think there's a lot of opportunity there. But then we also have this point where these combined geochemical techniques are the way to go, and that we get the sense for how the quality is changing. So this leads us into Hurricane Matthew, which has been a major focus for the lab group, and we've gotten a lot of insight into these storms out of this one event and how they affect coastal carbon cycles. So with Matthew, and this is largely done by Jacob Rudolph's a PhD student in my group. With Matthew, we saw again that we have this massive slug of carbon that came into the system. So we're looking here at box and whisker plots of the entire estuary and Pamlico Sound in time series, so dates on the x-axis. And you can see this increase in the amount of material moving through the system quite a long time before we return to baseline, nearly just about 50 or so days. And then a shift in the quality based on the isotopes that suggests that we've got more of this wetland material in the system. So it's hard to visualize in these box plots, and so we begin to look at cross plots of how the DOC properties change. And indeed, what we find is that the majority of the estuary and the sound samples are captured between these river and wetland sources. So the wetland ends up being a very large influence. Now, this was a nice kind of visual way to begin to see it, but what Jacob did was he actually used a spatial analysis and developed a flood-based model. And so what we were looking at is a way to basically say, okay, let's connect the river to its adjacent wetlands. And can we quantify both the amount and the quality of carbon that's being moved in, and does it fit with what the kind of idea that we've had before? And indeed, it turns out that this this worked. And so what Jacob did is he created the rather sophisticated way of looking at this model. And the upshot of the model was that he was able to distinguish when the hydrologic connectivity occurred between after base flow and then into this storm flow. And it changed the way that we were able to begin to quantify the flux of this wetland organic matter into the coastal zone. And so here I'm just showing you two images that he created, references down here. I'm happy to share it later if you're more interested in the details of this. But the upshot of that is that we've got this, this model now that says that, okay, if we can actually quantify, go out and measure, say, the wetland source, have a representative sample, that of the river, and then build this physical-based model, we can get this spatial connectivity and this spatial control over the type of carbon that's coming into the system. So Jacob was able to actually do that as well and build this into his study. And he used a Bayesian mixing model with our geochemical tracers here. And it fits quite well to demonstrate that after the storm, by and large, we have this dominance of the wetland quality throughout the system. And the upshot for that, getting back into thinking that how the reactivity of this material exists, is that the estuary and the sound tends to keep in a state of CO2 source. So a lot of times we think of these coastal interfaces and estuaries, especially ones that are mesotrophic to eutrophic like the Neuse, as CO2 traps because they have a lot of primary production and they're going to draw down CO2. What we ended up learning is that these storm events drive this different type of material into the system. And because of the nature of the embayed lagoonal estuary that is the Pamlico Sound, that retains that material for a long enough time it's processed, CO2 is generated, so on and so forth. So, now we're looking at here, what are some of the effects on the annual budget? We can see that this one storm was a very large amount of the material coming into the coastal system in 2016. And then we can also see that the wetlands were indeed the source of this reactive material. Our geochemical evidence kind of backed up this idea that we have this sustained CO2 efflux. The other point I'd like to make here is that we have a connection between a lot of carbon respiration and CO2 efflux that we can map up together. And these are some measurements of carbon respiration that Ryan Pearl's done, comparing kind of similar dynamics of Hurricane Matthew to Hurricane Florence in 2018. So it looks like this material is very microbially active. And so that kind of gives us, again, another indicator that what we want to do is to have a better way of describing and modeling this connectivity, change in source, and the movement of this wetlands into wetland material into the coastal system. So we're almost done here. And the third example we're going to talk about are seagrass communities. And so seagrass communities are something that another PhD candidate in my group, Cindy Labras, is working on. And this is nice because what she's doing is she's actually taking and doing a study in St. Joe's Bay, Florida, and looking at presses of urbanization, sea level rise. And the idea here is, how is the coastal carbon cycle and this very effective coastal carbon trap in terms of seagrass beds being influenced by some of these processes? So seagrasses are threatened by the presses of urbanization and sea level rise. You're changing the landscape. One of the things that happens is that turbidity ends up being a very powerful detriment to these ecosystems. And even storm events, as shown here. This is actually in the lower part of St. Joe Bay. After Hurricane Michael, can dramatically alter the water quality and then impact the system. So what Cindy's done is she's taken the spatial perspective using remote sensing to look at Landsat images and actually do classification study to translate what the satellites can see into carbon storage, into this below ground carbon storage here. And so the upshot of this one is to create these maps, but then to go back in time using remote sensing and look at a time series of seagrass area and develop this idea of the below ground organic carbon storage here. And so over time, one of the things she found, which was kind of surprising to us, is that for this study, that over 30 years, there's no significant change in the carbon storage. And so it does look like, even though these, we think these seagrass meadows, these ecosystems as being quite susceptible to being disrupted, they have a fair degree of resilience, and they're fairly stable carbon stocks. So just to wrap this up here, I'm going to try to pull together this coastal carbon perspective as to where we are, and I think you know, where we can go, and to try to tie this up. And so one of the things I want to point out is that when we look at the impacts of these storms, we find out they can be very significant amounts of carbon that's released and reacted and transferred. And so when we look at Hurricane Matthew, the effect of that was about 0.2 teragrams of CO2 released. Irene was about 5 times that size. A current model of CO2 release from eastern US coastal environments is the air to water CO2 flux of about 2 teragrams per year. And so we can see that these events are very large components of that. And they're actually not captured very well at all in these types of models. And so we do think we're making some progress with the work that we're doing, trying to understand more about how these events can be episodic, yet very significant amounts of carbon flux. And so we've sort of come up with this idea of the spatial context then, is that increasing the flood frequency shifts, you know, our major source of DOC from, say, a river, kind of the upland sources, into capturing these adjacent wetlands. And that one of the outcomes of that is actually pivoting where we see the CO2 or the system being a CO2 sink and switching it into a CO2 source. Now, in terms of looking at how the interface might reset or come back to kind of its original baseline, this is recent work from Eros Mala, who was visiting with Hans Perl and I and Ryan Perl in 2019. And he did this nice study that basically shows that this return to baseline is governed by mixing, but what can often happen is that we overwhelm that estuarine filter. And so some parts of the system, say the Neuse River Estuary itself, becomes more like a river and just delivers that material to the sound. where it has a longer residence time, and then the potential for retention and biogeochemical processing. And so I'll just summarize here with this little conceptual model that says that, you know, these storms can shift these coastal ecosystem states. And the big thing that, again, that we're learning is how the inputs are variable and the inputs are changing. And we are trying to develop better techniques of actually quantifying those changes and representing them more in what the coastal environments do. So I'm going to stop here and just leave you with the summary. And thank you for your attention despite the technical difficulties, and I'd be happy to answer any questions.
Yes, and everyone, you can feel free to, if you want to unmute yourself to ask, or you can use the chat. We have time for a couple questions. So Doug Newcomb says, have you looked at using the red edge bands in Sentinel-2 to map DOC in the rivers and estuaries over time, pre and post event?
We haven't. I think that's a really nice suggestion, and something that something we can certainly look into. I've spent most of the time trying to work on, you know, using ratios of the blue to other bands to try to quantify the amount, and we're beginning to use those to look at the quality. But I think this suggestion as well is a nice one, and we can look into that. Thanks.
Anyone else?
This is Carl Wegman. Chris.
Yeah.
Can I ask you a quick question? I'm curious about
Sure.
the seagrass, and just since it's, at least for myself, running around in terrestrial environments, not something I see or think about much. How, in terms of, can you give me a sense of kind of comparison? How much carbon does a seagrass contain versus kind of an equivalent size terrestrial plant? Is it the same, more, less?
Okay, so in terms of the carbon that it contains, it would be similar to other grasslands,
Okay.
I would think. And if there are any more vegetative-minded people there that know the answer, please do chime in. But the significant thing about them, though, is like if you think about their below-ground carbon storage, so like the soil, I mean sediment, it's not
Right.
soil, but the material that's built up there is enormous and quite an important stock of carbon. And so one of the other issues, of course, with this is that disrupting that system is going to release this carbon back into the environment. In a very similar worry that we have for how thawing of permafrost is releasing stored peat and Arctic carbon. So we're talking about, you know, spatially compressed areas, but very, very high amounts of carbon.
Right. Okay. Great. Yeah, that helps.
Okay, well, thank you very much, Chris. Really appreciate it. And I just want to remind everybody that there is a post-forum social, and I am, let's see here. There is a Zoom link in the chat window if you want to attend that, starting now. And again, just want to say thank you, Chris. Great presentation.
Thanks very much. Appreciate everyone's patience. I'll see you over in the social.
All right. Take care.
Thanks.